100 years ago, about 4% of the human population died from a pandemic. Pandemics are not a hypothetical thing. They're unlike many of the other dystopias that we've worried about from these other factors. They're a real thing, which led us to be probably both less dystopian and less utopian and much more practical in our layout of the various issues around these topics. So I will just summarize what some of the people who couldn't make it here had talked about. Dystopias from nanotech and biotech are actually pretty well known, as I say. Pandemics, bioweapons, terrorist attacks using bioweapons, accidental release of pathogens. These have sort of been well known. And in nanotechnology, unfortunately, some of the early attention to nanotechnology came about, oh my God, what could self-replicating machines do and eat the world? And that's eating the world is bad. So that's not new to most of the people in this room. However, there are some more practical applications that are a little bit different from that. So we covered a lot of things. One of the things we covered as well is the new stuff that's going on in gene editing in terms of biotechnology. Of course, recent news that most of you saw about a baby's genome being edited with CRISPR in China, and the progress of man. And as pointed out in our session, since we wanted to go in the future, by the timelines we're talking about, CRISPR babies will be doing CRISPR on their own babies to improve them even more. Seb, who is not here, was the most afraid of these bioattacks and of things like a weaponized Marburg virus or other things, as well as, in the opposite direction, master race biology. Countries starting to say, let's mold our population. Countries starting to say, perhaps that if you're in China and you'd like to have a boy, they used to just say you could only have one child and everybody worked. But if you'd like to have a boy, no problem, you can have a boy as long as you insert this into the gene line of the boy. Now, on the positive side, the story is also, I would think, actually reasonably well known to the audience here. The story of curing all disease, the story of manipulation of matter to the point that you can manufacture things at vastly low prices and remove the old problems of scale and cost and complexity in manufacturing and even new types of chemistry. These utopias are also well known to us. I would like Jun to have a chance, though, to cover some of the issues that she focused on, since you actually are here. Thank you very much for being here. Do you have a microphone? There's one right there by your leg.
Perfect, thank you. That was a really good summary of some of the topics we discussed, and thank you all who joined in the conversations about the bio track. My name is Jun Axup. I'm the scientific director and partner at IndieBio. We're a startup accelerator in San Francisco here. We funded 94 companies across all areas of biotech. Life as biology is super important because obviously if we do not exist, then nothing we talked about this whole weekend really matters. So there is the existential risk that we as a species may die, and not only is it due to the technologies that we might be creating, but also due to things like global warming that we know is getting worse, and if we don't step in to do something, that is going to kill us. Environmental manipulations, disease, and then of course just extraterrestrial things that we do not know about, maybe an asteroid will hit or something. Thousands of years of evolution, or millions, billions of years of evolution have really brought us here, and it's really interesting time for us because our biotechnology is able to actually change our DNA. So very much the most prime example in recent history is the designer babies. And while some people are excited about this prospect and other people are saying this is irresponsible and that we need more technology, this type of thing is going to happen. And while a lot of it is going to be trying to alleviate diseases, we also see that there can be eugenics problems, there can be more discrimination and more class dissociation, as the rich people are able to get these treatments and get these gene edits, and not the poor. But what I worry about in editing our DNA is that we actually might start selecting for certain traits that we want that are more favorable, and thereby decreasing the diversity of the DNA in our population overall. And this is very dangerous because if something catastrophic were to come, then we could potentially annihilate our entire species, right? And we've seen that in different countries, and we've seen that this could be a huge issue. And also from the philosophical factor, we could be decreasing in creativity and the development of new technologies by homogenizing our population.
Now this comes, of course, because I was really bad during the panel, and I kept pushing everybody, be darker, be darker. Where's my George Romero movie? Because we actually started out pretty tame, and I think now we've gotten into something that's a little bit darker. But it is, as I said, so pandemics are not like something we invented. The risks of biotechnology are certainly well known. Nanotechnology is much less understood. But you had some positive things to say as well. So,
yeah, definitely. We can definitely talk about how we can go about this. I just want to add one more thing, is not only is our human genes being edited, but we are looking at ways to change the environment as well. So there's gene drives that are trying to annihilate mosquitoes, West Nile virus or malaria, and it's a really interesting global, the ability for biotech to do that kind of thing on a global perspective, but understanding how that environment affects everything else in our ecosystem. We as humans are known to kill all kinds of species, and that has had devastating effects on the ecosystem. So we are very much playing God, and so should we be allowed to do that, and how do we do it in the right way is a question. And so some of the ways that we talked about more on the bright side of how we should go about this, a lot of it I think comes down to regulation and conversations and collaborations, just like what we're doing here, to try to address some of these issues and try to get everyone involved. I think one thing, especially in biotechnology, one of the biggest problems is this huge disconnect between the public perception and the scientific group, and that has led to a lot of misunderstanding, and of course just the lack of science education and the politics around certain things has made things very difficult. And so so I think a part of the fundamentals is having better education, getting the public to understand all the different consequences, and not just have one scientist make a decision, but to bring in everyone from all kinds of different sectors to have these conversations and to make these decisions more collectively. Some other ideas that we've talked about, one is, this one's kind of weird in that we might want to create defensive silos. Talking about if we do homogenize our population, and the fact that air travel is so common these days that if one virus comes about, it pretty much spreads around the globe in a matter of days. Should we actually be building up ways to physically silo people so that if one population were to be, affected by something, that we can keep the whole humanity still alive? So that's a little controversial, but I think that with the what was talked about in the neurotech session around better communication and more over the internet communication, we could potentially make that a possibility. And in general, we had a lot of talk about defensive technologies. So as there is a lot of fear around bioterrorism and bad actors, we should also be parallelizing ideas to try to defend them. Obviously, you can't anticipate everything that's going to happen, every molecule that someone would want to inject into the water stream, but having the technologies where you can identify problems and readily change them, like make antibodies to sequester certain types of viruses, would be worth investigating in. And then I think the more central side kind of wraps up all of this is that we really need more money in funding biotech. Right now, funds are going into very specific things. It tends to be curing cancer, curing Alzheimer's, which are great, absolutely big problems that we need to solve, but we can't continue to follow the trend and only invest in those things, because like CRISPR started out as a basic technology where some scientists were just observing the way viruses were being destroyed in a bacteria, and then came up with this technology, CRISPR, that is now applied to everything else in the biotech space. So if we don't invest in those basic technologies, we're not going to develop the next generation of tools that both will create cures and create and solve problems in our environment, as well as create those defensive cures as well.
It's okay. So indeed, this talk is going to be very heavily centered around the role that these instruments, often referred to as microscopes, they're not classical microscopes, they're scanning probes, the role that they've played in not only documenting nanotechnology, but also now in creating nanotechnology as well. So we're going to be talking about the materials that have gone into creating nanostructures as well as the instrumentation. I'm going to end up by talking about some of the newest commercializations of this line of instrumentation. It's not stagnant at all, it's very dynamic. Here's pictures of 4 happy users of these instruments positioned in front of 5 of their instruments. Most of the experiments being done in this area and in development, research and development, do operate in air. So if you have a preconception of an ultra-high vacuum, ultra-low temperature instrument, please wipe it away and realize that most of the experiments being done these days are being done under ambient conditions. The way that this instrumentation works takes advantage principally of very fine motor translation stages, typically piezoelectric driven. A tip, for example, in scanning tunneling microscopy will scan over a surface and measure scanning tunneling current. That tunneling current defines, the width of that tunneling current defines the resolution of this technique. In this particular image being shown here, each of those molecules lined up in those arrays is about 1 nanometer apart. There we go. So far more instruments have been developed along the lines of atomic force microscopy than scanning tunneling microscopy for various technical reasons. Atomic force microscopes, the resolution is limited by principally the contact area between the tip and the sample. The tip is typically silicon, and the radius of curvature is around 10 nanometers. And so the resolution of the data that comes out of this instrument will be 10 nanometers or so, as opposed to the 1 nanometer that you saw with the scanning tunneling instruments. Okay, so what brings me first in touch, and I have to diverge a little bit here with Eric Drexler, was that picture right there. Shortly after we published this in Nature 30 years ago, 1988, I believe, we were in contact with Eric about, well, at that time it really was nanotechnology, molecular manipulation, molecular identification. And I use this just to show how the instrument really has been one of the accompaniers to nanotechnology in its ability to document molecules on surfaces over on the left, to document how molecules interact with other molecules over here. And what we're showing in the middle picture is a transition that this whole field made from being able to document just topography to being able to document actual mechanical properties. So this map on the bottom, which looks like islands and seas, that's islands of a few nanometers in a cohesive sea. They aren't actually physical items. They're rather areas of differing friction. And so we're able to configure instruments to measure friction across samples, particularly when you have mixed materials. So you can see on what length scale mixtures of materials are now phase separating. So often now in nanotechnology, these instruments are invoked to measure nanoparticles and to measure nanopores. The way that you read these data, which are rendered here by a kind of, almost looks like ray tracing, is that light is high and dark is low. Nanoparticles on the left, it's a little bit not homodispersed, but they aren't that far out of a range. And on the right, you can see nanopores, which are fairly more homodispersed. In fact, they exhibit a hexagonal close packing. Now, I was pretty kind to you in the way I presented the slide to you. To my colleagues, I would show it in this manner. I would take credit for chemistry in saying the reason why we have homodispersity here is due to the chemistry. And both of these structures resulted from the same mixture of 2 materials. In fact, 2 polymers, one which contains silicon on the right and one which does not contain silicon on the left. So why does one give particles? Why does that same mixture give either particles or pores? Well, it's all in the chemistry, my dear. It depends on what solvent this is being processed from as to whether the continuous phase ends up being the derivative of the silicon-containing polymer or whether the particulate phase ends up being the derivative of the silicon-containing polymer. And we're not going to go into that chemistry right now. Lucky you. But the point is that the chemistry is really what's a sensitivity of the chemistry and the choice of the materials, and then in the processing of it is what gives us control over these nanostructures. And control is really important. In fact, that would have been one of our take-home lessons in our conclusions from yesterday's discussions, is that this is what nanotechnology, the good news and the bad news about nanotechnology is control. And the way that we get control in many of these nanostructures is from the actual molecular structures of the starting materials, which more and more these days are polymers. Polymers are sequences of monomers, chains of beads. And when you have mixtures of polymers which can't get apart from each other, they're covalently hooked up, they will phase separate in these idealized structures here shown as spheres, cylinders, lamellae. And because these different chemical parts will have different chemical reactivity, once they phase separate, we can remove one of them. In this case, we're removing the continuous phase, and what we're leaving behind is the particle phase, the nanoparticle phase, the spherical phase. And that phase is particles all of the same size. And so we have control over the size of the nanoparticles here. Where did that originate? Where did that come from? That came back from that wiggly worm of a molecule you see above that we call a polymer. The size of the sphere correlates with the length of the polymer, and the length of the polymer is very controllable by the synthetic polymer chemist. Likewise, if we have spheres arrayed in this manner and we dissolve the spherical phase, what we have left behind is the nanoparticles. Again, homodispersed, and those nanopores are the size they are because of the original polymer length, the molecular weight of the polymer. So once again, a control over uniformity, which is really what the challenges in nanotechnology are deeply steeped into, and that was one of our take-home lessons from yesterday's discussion also. Do you have control over this so that you can anticipate what properties it's going to have and direct those properties as well? Another one of the challenges of nanotechnology is, can you find them? Can you locate them? Can you put them where you want them to be, actually? And there have been several studies illustrated here of putting nano-objects in their places. What we see are two examples on this slide. One's of magnetic nanoparticles being trapped in a conventionally lithographed trough, and the other is of DNA triangles. I bring that in because biopolymers is one of the compatibilities between the two sections of our study group yesterday, between biology and nanotechnology, and that biopolymers can also be used to create nano-objects. And in this particular case, that dark continuous line is a single strand of viral DNA, which has been artificially folded and bound back by artificial strands, which are shown in color. In fact, they're multidentate, not just bidentate. And in this manner, Paul Rothman at Caltech was able to create all of these whimsical little structures, which are just about 100 nanometers by 100 nanometers. Cool, but what for? Well, if you can demonstrate that you can now take a biopolymer and reconstruct it into a shape which is of your advantage in building something else, and then you've shown being able to take advantage of nature's binding properties to create artificial structures. So you create them, but once again, you want to find them, you want to place them. And the example being shown here is of an electron beam lithograph surface. That's the series of what looks like triangular holes there, onto which you cast a solution of these DNA triangles, about 100 nanometers on a side. They dock into or onto those landing sites. And here's magnified a bit. You can see that not only are they docking one by one, but they have an orientation as well. Now, in fact, this surface doesn't have holes in it. It's been rendered as if it has holes in it. What it really does is have a patterning of surface energies or chemical species where you have greater attraction to where the triangles are docking. I'm not going to go into that right now. But once again, you can say, cool, but what for? Well, think of the chip. Think of a board and think of the chips that sit on it. And now take that down 3 orders of magnitude in size and think of these DNA origami as being chips. Now, how would they become functional chips? Well, we know the sequence of the bases in the DNA, and so we can then functionalize it. And in the case where you functionalize the sulfur, or you functionalize with sulfur at particular sites in that DNA structure, you can now bind gold. Gold and sulfur have a particular affinity for each other. So on exposing these sulfur derivatized DNA origami to gold nanoparticles, you can position the nanoparticles intentionally in space. So this is partly about showing control over matter, going from DNA constructs of 100 nanometers now down to gold nanoparticles of less than 10% of that. Now, will this go into technology? Maybe. This really was more study and demonstrating using biopolymers to create nanostructures. But what it's also doing is showing that perhaps the biopolymer will be a temporary phase. It'll just act as a scaffolding. It'll be sacrificial, and you'll burn that carbon away and leave behind the gold particles. And then you'll do some interconnectivity between them. The good news and the bad news of nanotechnology or nano objects, the good news is it's just so terribly localized to a few nanometers. But the bad news is it's so terribly localized to a few nanometers. If you're going to manufacture with some of these tools, you have to think in terms of how am I going to scale this up? Otherwise, it's going to take too long to build on this small scale. And one of the schemes for scaling up is to build these vast arrays of these tips. And in this particular case, I'm going to play a movie here because I can. It was made by IBM Zurich Research Labs. And what we're seeing here is a scheme they came up with about 10 years ago to use these force microscopes as actually data storage systems. The tips were heated and they were capacitively pulsed into a plastic sheet. If they made a hole in the plastic sheet, or where there were holes, those were ones, and where there were not holes, there were zeros. This was nicknamed millipede, and it was one of the first devices based on nanotech to show that there's a demonstration that one could go forward in building in this, on this length scale with these instruments, or at this length scale period. Now, each of those tips is independently accessed. Each is an independent circuit so that you can operate them independent from each other. That is, you can make a 1 or a 0 independent of each other. Swiss watchmakers in the 21st century. It's wonderful. Okay. So did that come to be? No, it did not come to be. It was not competitive with flash. But the success story is that it did demonstrate the capability. And the way that that capability has been taken on has been with a single probe in order to create, for example, single masks. And once again, polymers come to the rescue, designer polymers. A chemist would say this is self-amplified depolymerization, whereas an engineer would say it's one-step dry lithography. And what's happening here is that we're etching down into the polymer with a heated tip and causing that polymer to depolymerize or vaporize so that the material is disappearing. And in that way, in one step or multiple passes, you can actually etch down into a surface at different length scales. There's the 1 micron scale bar. Remember, dark is low, bright is high. So this is fun. This is another material that also volatilizes when you bring a heated tip into it. Perhaps you recognize that morphology. It's the Matterhorn. Once again, this was done at the IBM Zurich laboratories. And once you do this in polymer, you can then use it as a classic resist, and you can transfer the pattern into silicon for those of you in this semiconductor domain. This does not need an explanation. It's taking that previous Matterhorn. Yes, the Alps, if you know where Switzerland is, you'll see the Alps that are part of this map. Okay, the very last things I want to mention is where we're now taking these instruments into being very active, not just imaging instruments, but very active in creating structures as well. In this particular case, the AFM cantilever was hollowed out, as was the tip, to create a delivery system. And originally, Cytosurge, the company out of Zurich, it's a spin-off from the ETH, intended to pierce cell walls and to introduce components into cells. But how this has come to be used by, I think, many more people is as a nanofluidic delivery system. The tip, here's an SEM of that tip. You can see it has a 300 nanometer hole on the bottom. Does that mean that we can now make features of 300 nanometers? No, not at all. The fluid dynamics take over once you release fluids from a hole that size, smaller or larger, and the size of the resulting features really are more a function of capillary forces and surface tension. In other words, all bets are off until you do the experiment. And the experiment's been done using, again, this high precision stage, which developed with AFM and this nano delivery service to create arrays. Now, what's really remarkable in this slide, this is the concept. You can see there's a millimeter. You can see there's a millimeter scale bar up at the left. So once again, the downside and upside to this technique of manufacturing on the nanometer scale is that you do things so localized, but you need to be able to scale up if this is ever going to become feasible in, particularly in additive manufacturing capability. And these SEM images here, which show these lines actually being composed on top of each other, have enabled us to document that the repeatability over 1 millimeter of being able to deposit subsequent rows on top of each other is 4.8 nanometers. Now that's really incredible. It requires software correction. Why is it incredible? Because thermal drift on this scale is much larger than that. Another downside to trying to do this kind of nano construction is that KT surrounding us is a killer, or can be a killer. So it's something that you have to be very conscious of and use software correction on. Okay, another, the same instrument, like I said, it isn't, I don't think it's piercing as many cell walls as it is being used in additive manufacturing. And in this case, it's creating a copper coil, a copper helix that can't easily be created using other techniques in this 3D array here. You can see the scale is about a micron or so. It was configured as an electrochemical deposition to deposit metallic copper from a copper sulfate solution. Okay, I think this is a finally. Yes, this is a finally. The AFM tip, as I mentioned, has a radius of around 10 nanometers and gives exquisite resolution topographically over 10 nanometers. But one of the Achilles heels of this technique is you can see topography, but the chemist in you, or the chemist in some of us, needs more information than topography to really nail what the molecular species is. Well, there are now advances in this instrumentation that brings infrared spectroscopy into this tip sample interface in order to give us AFM images that are coupled with infrared spectroscopy. So in this particular example, we have one of those lamellar phases of a block copolymer of polystyrene and polymethyl methacrylate, and we're able to detect very different infrared signals from those two domains. In other words, it's not just topography anymore, but it's also now getting a fingerprint, a thumbprint of the material that's under the tip. So in that way, we can take our nanoparticles now that can be core shell nanoparticles, meaning a polystyrene core with a polymethyl methacrylate shell, which is what we have here, just about 10 nanometers in extent, and be able to distinguish between the shell, which is being shown here in red because it absorbs at 1720 reciprocal centimeters, that's polymethyl methacrylate, and the cores are showing up with the frequency or the wave number for polystyrene. So we can now chemically distinguish between different components on this scale, which is a big step in this nanotechnology field, to be able to analyze just where each of the two materials is located. So this is a slide which was made by my colleagues for me last year at an award assembly. This is, I think it's the smallest sculpture known. It's 1 micron by 1 micron. It's been etched into silicon using an oxidative process. It was done by my colleagues at Asylum Research in Santa Barbara, and it's silicon oxide on silicon. And I want to thank you for indulging me in this little educational session today.
Thank you.